Hello. So we should still be on day one. You have initially looked at the intro to tell you how to get signed on to the course. And then we did a review of some basic cell biology topics. Now we're going to move on to genetics and some of the causes of genetic disorders. This is not a genetics course. You may have already had some genetics, but everybody needs to know enough about the processes of gene splicing, reproduction of genetic material, so that you can understand how you can have genetic disorders, congenital disorders, meaning that you're born with them. First of all, we're talking about eukaryotes here, because we only care about genetic disorders for people. And so we have a couple of different kinds here. Meiosis is cell division between gametes, in other words, between a sperm or egg, in which case the reproduction of this would leave you with just half of your genetic material. Creation of an egg, you're going to have 23 chromosomes, not 23 pairs, because you're going to pair up with the 23 from the sperm. So cell division in gametes is called meiosis. Mitosis is cell division in every other kind of cell, in which case you have to replicate the entire 46 chromosomes, or 23 pairs, so that each cell winds up with 23 pairs of chromosomes. Cytokinesis is the process of this, the whole process of reproduction of the cells, the breaking apart. It means cell movement, actually. So we're looking at our DNA strands that are packed into these chromosomes. The DNA strands have, thousands and thousands and thousands of pairs of nucleic acids, which make up basically the alphabet that spells out what's going on in your body. Small sections of those are genes. A gene just for your hair color might be contained within this chromosome, which is, a long piece of DNA. These DNA strands, it's a double helix. They're twisted around each other, and they're held together with hydrogen bonds, which are very weak bonds, so it's easy to split them apart, reproduce them, put them back together. We have two pyrimidines, two purines. We have cytosine and thymine, adenine and guanine, and those are the only four that we're using to make these. At its most basic, it's almost as simple as a computer, where you either have a 1 or a 0, but you have so many of them that you can code for millions and millions of different things. The cytosines and guanines will always connect together when it works right, and the thymine and adenine. TNA binds together, and the cytosine and guanine will bond to each other. So there are a couple of more things that will happen here. You can either split the entire DNA strand, reproduce the whole thing if you're splitting the cell, or you can just split a part of it apart, make a copy of it here, and make that protein that you need. So protein synthesis is going to be a big deal here. Either a problem can have when you're reproducing the entire DNA strand, or it can happen when you're producing a particular protein. For instance, one that might be a collagen protein in Marfan syndrome, where you're unable to produce the collagens that make your blood vessels strong. So we're synthesizing proteins. A protein is made out of amino acids. They form into peptides. A dipeptide is 2 amino acids. A tripeptide is 3. Polypeptide is more. You take a few polypeptides and stick them together, and you have a protein. The protein is your actual functional unit that causes something to happen. DNA replication as well as protein synthesis. But DNA replication, generally speaking, you have to untwist this tightly coiled DNA strand so that you can then split it apart. These DNA strands are so tightly coiled that although they're microscopic, if they were actually completely stretched out, they would be about 6 feet long. So they're huge, 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 but they're very compactly rolled. So you have to untwist them, then you have to unzip the part that you need. Then you get a complementary base pairing, these RNA, pieces that are made in the nucleolus will come along and they will match up with the DNA strand, basically forming a mirror image of the piece of that DNA strand that you want. The base pairing is done correctly and is controlled by an enzyme called DNA polymerase. Making a polymer means to put a bunch of things together. So DNA polymerase, if it's working properly, will make sure that this matchup is correct all the way from one end to the other. So here we've unzipped. First of all, we uncoiled it so that it's a loosely coiled structure. We've unzipped the pieces here, and now we're sticking on the complementary base pairs, the complementary RNA chunks, to form this piece of the DNA that we want to replicate. The DNA polymerase is marching along here, making sure that A goes on the T and the C goes on the G, and the C goes on the G and the T goes on the A, so that everything is correctly lined up. Mutations then are very possible at this point, because if you leave one out, you have a mismatch somewhere. If you put the wrong one on, if the polymerase doesn't do its job and you match up the wrong two, you're going to have a defective piece of DNA. And if you have a frame shift, if you leave one out but then the next whole piece of DNA moves over a chunk, or if you put an extra one in and it moves it this way, the whole rest of that DNA strand is frame shift in one off. And so that piece of DNA won't work properly. As you can see here, if this one was missing and the rest of it just moved right into spot, then every one of these would be misaligned. Mutations occur every once in a while spontaneously. If the mutation's bad enough, the cell will die, and then you don't have a problem, presumably, unless the cell manages to live and reproduce that mistake, then suddenly you have a additional problem. Spontaneous mutations are just ones that happen every once in a while over the billions and billions of these things that you have in your lifetime. Or it can be encouraged by a mutagen, such as a carcinogen, something, or thalidomide that cause people to have birth defects, kids to have birth defects. Mutagens cause these mutations in the DNA to happen. Sunlight, UV radiation is a mutagen. It causes DNA damage and DNA mismatches in your skin and causes skin cancer. Mutational hotspots are places that have been located where it's uncommonly common for a mutation to occur. So if all works right, you're making a protein here. Let's say this codes for the protein. So you split this apart and you bring in your RNA, your matching nucleotides. You put them on there in the right order, and then you take this piece off, and again, you have what is a mirror image, because you had AGACG, now you have UCUG, and so what you have is a mirror image of what you actually want. That's called transcription. It happens first. It happens on the DNA strand inside the nucleus. It just untwists and you get this piece. This piece will move outside of the nucleus. It will go to a ribosome, which has the ability to now match up corresponding pieces onto that mirror image, and you'll produce that, and when you pull it off, it's the mirror image of the mirror image, so it's the piece that you originally started out with. This is called not splicing, gene splicing. Gene splicing, where you line these up. Here the piece comes out here, it moves past the ribosome aligns the AG, and suddenly you have a peptide, a polypeptide, whatever. These things go into the Golgi apparatus, which packages them into their final form and takes several polypeptides, puts them together, and you have a protein. So transcription takes place in the nucleus. This process is called translation. Transcription comes before translation. This translates your original idea into the final form. So that's good enough for knowing how that works, because breaking it apart, polymerizing it on, where you can have an extra one, not enough, or a frame shift to cause a mutation. It can happen here, or it could happen at the ribosome, either one, because there are two different places this can happen. That's good enough for you to understand where most of the mutations are going to occur. Now, these mutations can occur in a body cell. Soma means body. So a somatic cell is just a general body cell. The gametes, or germ cells, are sperm and eggs. So you're supposed to, in every body cell, have 23 pairs of chromosomes. Each chromosome is one 6-foot piece of DNA crammed into this tiny package. And so we've got 23 pairs of chromosomes, one from mom, one from dad, and that is going to carry all of our genetic material. The gametes will only have one set of chromosomes, so they have 23 instead of 46. The somatic cells, because they have two sets, in other words, 23 pairs, are called diploid, di meaning two. And the gametes are haploid. They only have half as much, so they are the ones that have just 23 chromosomes. Now, these chromosomes are divided into ones that do everything for you except tell whether you're male or female, and then the ones that decide whether you're male or female. The autosomes, those are myosomes, and they are the ones that will do everything. Hair color, your height, your eye color, everything. 22 of the 23 are autosomal cells. Autosomal chromosomes. Only the 23rd pair, one from mom and one from dad, are your sex chromosomes. So that's your X and Y chromosome. So any kind of a mutation up here will cause one kind of problem. It might make you miss a finger or something. A problem with the sex chromosomes will have a problem with your development as a male or female, or your ability to reproduce and that sort of thing. So any cell that has a normal nucleus, a normal looking nucleus with the normal number of chromosomes, everything being right, is called a euploid cell. Eu meaning good. Eustress is good stress. Euploid means they have a good looking cell and that has a normal number of chromosomes. So for a somatic cell, that would be 46 chromosomes, 23 pairs. For a gamete, it would be a single set of 23. Those would both be euploid because they're what they're supposed to be. If you have an extra entire set of chromosomes, instead of having 23 and 23, the pair, you have another strand. So you have, instead of 46, you have 46, you have 69. Then you have polyploid. You have too many entire sets of chromosomes. These, in tetraploidy, means you've got 4 strands. Basically, almost without question, fetuses don't survive having an entire extra set of chromosomes. They die. You probably never even know you were pregnant. You have a menstrual cycle. It's actually a miscarriage, but you don't know it because the fetus couldn't live with an entire extra set of chromosomes. However, you might have your normal 2 strands, but 1 chromosome of those 46 you have either is missing or you have an extra one. Now, fetuses will survive that. Instead of having 3 whole strands, you've got 2 strands but 1 extra chromosome somewhere, or missing that chromosome. And then you have aneuploidy. In other words, not good looking. And anything means not. So aneuploidy is a cell that has the wrong number of chromosomes by just a chromosome here and there. If you have an extra single chromosome someplace, instead of having, 1 on each pair, you have an extra one over here. That's 3. It's called trisomy. And like a tricycle, 3. Okay, so you can have an extra individual chromosome, trisomy. If you're missing one, you're supposed to have 1 on each pair here. If one of them is missing, you have monosomy. In other words, you only have 1. You're supposed to have 2. Usually, a fetus won't live if they're missing a chromosome altogether, although in some cases they can. They're more likely to survive if there's an extra chromosome at one location like that, because sometimes the more powerful normal chromosomes will dominate that one, and you'll at least get what you need to stay alive. It's better to have an extra one than not to have one at all, because without one you just don't have the coding of some part of your body that you need to live. The term karyo means nucleus. So a karyotype is a nucleus type. So here are some different kinds. You can have a euploid normal karyotype. That's what you want. But if you have a nucleus that has that extra chromosome here or there, or maybe even missing one, it's a different karyotype. Down syndrome is probably our most commonly known and not terribly uncommon to occur, aneuploidy. Not the right looking number. This karyotype, at 21, you have 23 pairs, right? At 21, you have an extra chromosome. And this is kind of a hot spot for a mutation type of thing, in that it's, 1 in 800 live births. That's, a pretty small number overall, but if you have hundreds and thousands and millions of births, you're going to have quite a few Down syndrome kids. Mentally retarded, the low nasal bridge, the epicanthal folds, which is the fold over the eye that makes them look a little bit different. Their tongue is too big and sticks out, makes it hard to speak. They have high cholesterol levels. They tend to die of heart disease relatively early. And the risk of having a Down syndrome aneuploidy kid with trisomy 21 goes up dramatically after about age 35. So here's a child with Down syndrome. And Down syndrome, all of these, it's interesting, they're not all the same. Down syndrome can be anything from a kid who can't even learn to speak at all to somebody who can make B's in high school or star in their own TV show. I remember, probably 15 years ago or something, there was a TV show on the series where the star of the show was a kid with Down syndrome, and he was going to high school. You can see 1 in 1,000 if you're young, all the way up to about age 35, it doesn't change that much. But people over 35 trying to have kids, women in their 40s trying to have kids, it can be 100 times more likely to have this genetic defect. Trisomy X, this is your X sex chromosome. So, here's Down syndrome. There are a number of them. And when we go through the different diseases and different body parts, we'll talk about different, or body systems, we'll talk about the various different types of trisomies that you can have. Couple of examples of the sex trisomy. This is a female that has a 3rd X chromosome. All the rest of her pairs are right. Instead of 2, though, she has 3 X chromosomes. And everything from moderately severe to super severe birth defects. Sterility, mental retardation, and if you have even a 4th X, it gets worse and worse and worse. Turner syndrome is a female that has everything else right, but only was born with 1 X chromosome. They tend to be really short, thick neck, undeveloped breast, undeveloped, usually infertile, or their fetuses spontaneously abort. Here's a young lady with Turner syndrome. And you can see this is her genetic makeup. You see pair, 5, 6, 7, 8. Only 1 X. She doesn't have a Y because she's not a male. And she only has 1 X chromosome. If you have an X and a Y, the Y makes you a male because the Y chromosome causes your primordial gonads to become testes and start making testosterone, and that's what turns you into a male. So if you have an XY, you're going to be a male, but if you have yet another X, you have Klinefelter syndrome. So basically you're a male, but you have some different type of additional female characteristics. Development of female breasts, very sparse body hair, sometimes mental retardation. So those are examples of sex chromosome aneuploidy. You can have other types of chromosomal damage. For instance, if you break a chromosome, when you're trying to make a protein and it breaks and then tries to get back together, there can be gaps, there can be shifts. You can have a piece that breaks off and doesn't work at all afterward. You can have the breakage. If you're trying to do 2 of them and this one breaks in half, they can shift and come back on reversed. So you can have a variety of problems with simply reproducing the chromosome where you shift pieces around or lose a piece. And here's again, all of these are uncommon, but these are common enough that they have given names. You'll probably run across them at some point. And this cri du chat, meaning cry of the cat syndrome, it's a part of chromosome number 5 is broken off and deleted. Maybe it never developed in the first place, or it broke off because it's a weak point in your chromosomal linkage and you don't have it anymore. And so you're born with microcephaly, small head, mental retardation, very short, pudgy fingers. And it's called cry of the cat because it changes your vocal cords, so when the baby cries, it sounds like a cat crying. Let's talk a little bit about the terminology of genetics, because we're going to be dealing with what happens if this chromosome or this gene is misplaced. It's in another place or location or locus. If you have a DNA strand like this and you have two of these coming down, and your gene has a gene from the father for brown eyes and a gene for the mother for blue eyes, that's the locus, is the location of the gene for eye color. And you have two loci. Now if this were put on a different locus, they might not match up well enough to work. So what you have is brown on one locus, blue on the other locus of the matching pair. The allele is the type of gene that's on there. So if you have a brown and a blue, you have a brown allele and a blue allele at the locus for eye color. Heterozygous means you have two different ones. You have a brown and a blue. Homozygous means you have a blue and a blue, or a brown and a brown. They're the same. You don't have two different genes. They're identical on your two different strands here. Again, some of the possibilities that you can have: a translocation means that the allele gets put in the wrong place. A translocation of a gene or a piece of the DNA. Inversion, when it reattaches, it gets put on like this. It is flopped and it's in the wrong order. Duplication, if you wind up with an extra piece. I've got a brown, I've got a blue, and I've got a blue. Now something isn't going to line up properly. Your genotype is your genetic code. My genetic code says I will have brown eyes. My genetic code says I'm a male. Your phenotype is what you actually come out looking like. If I have a genotype for brown eyes, but they come out blue, then there is something strange going on, but I will look different than I'm genetically designed to be. There are male fetuses that come out looking female because they can't respond to the testosterone they're producing. So their genotype is male, their phenotype is female. When you have brown and blue alleles at this locus, it's very common for one of them to be dominant over the other one. Brown is dominant, blue is recessive. So if you have one gene for brown eyes and one for blue, you'll have brown eyes, unless there's some other abnormality going on. So the dominant gene, if you're heterozygous, you have two different genes at the same locus on the two different pairs. If one is dominant, it's the one that will express, and you'll look like that. And the other one will be a recessive, and you won't look like that, but you'll be a carrier for that. You can be a carrier for blue eyes. So two brown-eyed people can have a blue-eyed baby. If they're both carriers for the blue gene, it doesn't show up in them. But when they have the baby, if they get the blue from both parents, then the baby has blue eyes. Dominant, whatever it is, the dominant allele or dominant gene is shown with a capital letter. The recessive is shown with a lowercase, small letter. So a carrier is somebody who is heterozygous. They have a dominant gene that is what they think they are, but they have a recessive gene that they could pass on to somebody else. A carrier is not expressing the recessive gene. If it's a gene for a disease, then they don't know they're sick. They aren't sick because they're not expressing the recessive gene for that disease. For example, sickle cell anemia is a recessive gene. So you would say, well, this person is heterozygous. They have one normal one and one recessive bad one. So this person would be a carrier. They wouldn't know they have sickle cell in their DNA. However, if you have both of the recessive genes, you'll have the disease. You will express that disease. You don't have to memorize all of these, but you should get kind of used to them so that you'll be able to at least look back on this slide if you ever need it and say, okay, I know what this genetic map is. If you're looking at a genetic map of a kid who has a genetic disorder, you may want to find out where it came from. This round is a normal female, square is a normal male. The diamond shape means it's just a person. It doesn't say whether they're male or female. Apparently it doesn't matter in that particular thing. Single, here you got a male and female have mated. Here you have a male and female have mated, and they produced 3 normal kids, 2 girls and a boy. Here's a single parent. You're only looking at that parent's genetics. We don't care about the other. Maybe we don't even know who it is. So here's a parent and their 2 girls. The double bar means consanguination. This is marriage of close relatives. It means sharing of blood. So brother, sister, first cousins, father, daughter. I mean, this happens with, especially with sexual abuse. You wind up having a baby with 2 close relatives. It's bad because they're more likely to have the same genetic problem and pass it on to the kid. So very quickly, genetic abnormalities grow in consanguination. Here we have fraternal twins, a boy and a girl over here. Here we have identical twins. They have to be the same. 2 girls, 6 boys. The darkened circle means they have the disease or are expressing the disease. So here's a sick girl and a sick boy. Here they're dead. A carrier has the dot in the middle, so they don't have the disease, but they're a carrier for it. So go through this a couple of times. Another term that's useful is the proband. Usually the dark square will be the proband. This is the first person in that family to express that disease. So if your grandfather is the first person in history that you know of anyway that showed cystic fibrosis, then that person is the proband. And those going down from that person will be more or less likely to inherit that disease, depending on where they fall in this genetic layout. So, if you have gene disorders, and we're not talking about whole chromosomes, trisomy or monosomy, we're talking about within a chromosome you have a gene. Now we're getting pretty specific, but it could be a gene for sickle cell anemia. And that's bad. You know, it could be a gene for cystic fibrosis or multiple sclerosis or something. So anyway, either you can have the gene that's bad could be your dominant gene of a pair, or it could be the recessive gene. If you're heterozygous and your dominant gene has a disorder, then you're sick. If you're heterozygous and the recessive gene has a disorder, then you are a carrier. You're not sick. You may not know you have that gene at all. So if the abnormal allele is dominant and the normal one is recessive, they're on autosomes, so they're not sex chromosomes. These are regular body ones, so they will do things like eye color and hair color. Then what you want to do is to establish the recurrence risk. If I have a recessive gene for such and such a disease, and I want to have kids, how likely are they to get sick? Well, it partly depends on whether my spouse has that same recessive gene or not. So depending on what you have and what your spouse has, there's a certain likelihood of your children having that disease. So recurrence of an autosomal dominant, now remember, if you get this gene, you're sick because it's a dominant gene. I have one dominant bad one and one normal good one. Well, my kids are 50-50 chance of getting this one and this one. So your chance of your kid getting that disease is 50-50. Consanguinity is more because you have more likelihood that from the same proband you both have some sort of disorder. Maybe the other one might have a recessive disorder that adds to it. So here's the way we do this. Autosomal dominant. So the D is the dominant bad guy. The little d is a recessive okay guy. Remember, this is autosomal dominant. We're heterozygous. Heterozygous is the parent here. That means you've got one bad one, one good one. Hetero means multiple or different. So here we have two affected parents. This one is heterozygous with the dominant gene. This one is heterozygous with the dominant gene. Both of these are sick. They shouldn't be having kids. But maybe it's a disorder that is very mild. Or maybe we're just talking about hair color here. I mean, it works the same. But since this is pathophysiology, the likelihood of having brown hair is not a disease. The likelihood of having cystic fibrosis is. So we'll be concentrating more on the risk of having this actually show up. Well, so here's mom, here's dad. Well, the chances are 50-50 that, you know, half your kids are going to get your bad gene and half of hers are going to get the bad gene. So a quarter of your kids, 25%, will have both bad genes. If it's a serious disease, they should never have children. They should adopt, because every child this person ever has will have this dominant disorder and will be sick. It's a dominant gene. They can only give a sick gene. Now, some will get her good one and his bad one. They'll still be sick, right? It's a dominant disorder. But at least they only have half the genes. They could presumably maybe have a kid that wasn't sick. Same here. The bad one here, the good one there, heterozygous, affected though because it's dominant. And only 25% of your kids would get the good one from each one of you and not be sick, not be a carrier, not be anything. Okay? So you have 25% are doubly bad, 25% are okay, and 50% have one of the bad genes, and therefore they're sick. If somebody with this dominant trait marries somebody, has a baby with somebody who doesn't have the trait at all, then half will have the bad gene. They will be sick, but they will be heterozygous. The other half will be normal. They won't have any of the trait at all. Autosomal recessive means that in your normal, non-sex genes, you have a recessive trait that is faulty. Now again, it could be for blue eyes, which is a recessive trait. It would work the same way, but we're talking disease here. So the normal allele that is dominant. The big D now is the good guy. Your recessive gene is the bad guy. So let's see what this is going to look like. Cystic fibrosis is like this. Cystic fibrosis is a gene to code for a chloride transporter in various tubes of your body, but especially in the lungs. If you can't move the chloride, you don't move water by osmosis, you can't thin out your mucus. The mucus gets so thick you have to beat the kid on the back to get the mucus to come out. Cystic fibrosis is a autosomal recessive gene. So you might give it to your kid if you don't even know you ever had it, because you might be a carrier for this. You can't be a carrier of an autosomal dominant problem because you have the disease. So you're not a carrier. A carrier is somebody that doesn't know they have it or isn't expressing it, but it's in their genetic makeup anyway. So here we have, let's say we have two parents and they're both carriers for cystic fibrosis. Now the big D is good, the little d is bad. If you get the two big Ds, you're great. You're homozygous normal, you will never pass this on to a kid. If you get one big D and one little d, you're a carrier. Remember, it's recessive. So you'll be a carrier, but you won't have the disease. But you can give it to your kid. And 25% will get both the bad ones, and you'll have cystic fibrosis. So in this case, 25% are normal, 25% are affected, and 50% are carriers. Practice some of these. You're going to have to do it on the test, so practice some of these. There are other ways, other diseases, that are a combination of genetics and lifestyle. Now, total lifestyle ones, we'll talk about those too, but we don't care about that right now because we're talking about genetics today. But there are a lot of diseases that have a combination of effects, a multifactorial effect. Polygenic. Genic means to create. So a lot of different multifactorial things. So you can have, for instance, breast cancer. If your mom had breast cancer and your sister had breast cancer, you have a much higher likelihood of having breast cancer. But also breast cancer is affected by hormone drugs you might take, birth control pills, obesity, diet. There are a variety of things that can also add to your likelihood of having breast cancer. Heart disease is a good one. If your father had heart disease and your mother had heart disease, your chance of having heart disease are very high. But you could have a bad genetic background and take great care of yourself and not wind up with heart disease. You can also have nobody in your family with heart disease and treat yourself like crud your whole life, obesity and smoking and high alcohol and no exercise and all this kind of stuff, and wind up with terrible heart disease. So it's a combination of ingredients in this case. The threshold model says that, well, you have this factor and this factor and this factor. If you add these up, there are like 10 different factors that go into predicting heart disease. If you have more than a couple of those, your chances go way up. If you have several of them, your chances go up even more. So your recurrence risk is based on lifestyle and genetics. Now we can look at your lifestyle and make sure that you diet and exercise properly. That will help. But also looking at your genetic background, your family history. And this doesn't mean my dad came over on the Mayflower. Family history, my dad had heart disease. Okay, so if one close relative has had heart disease, my father and my brother, then I'm more and more and more likely to have heart disease too. If the proband, that first person that showed heart disease, had it really, really bad, had 2 heart attacks before the age of 50, your risk is higher. If it's the person that isn't usually have heart disease, if your mother had 2 heart attacks before the age of 50, that's even worse than your father having it, because she should be less likely to have it. And it decreases very rapidly if you get farther away. I mean, if it's your grandfather instead of your father, then you have far less risk. If it's a cousin, way less risk. An uncle, less risk than if it's a father or brother. And we can do genetic mapping to determine your overall risk of having these various types of diseases. And one of the most famous nowadays is, we call it the BRCA gene, although it's BRCA, breast cancer 1 gene, BRCA1 gene. Women who have that are at a much higher risk of having breast cancer. A good friend of mine, the wife of my best friend really, has had breast cancer twice. And it was 2 different kinds of cancer and 1 in each breast. So she went and had a full genetic profile done. She not only has the BRCA gene, but she has like 8 other genetic markers of breast disease. So she had both breasts removed prophylactically, so she won't have breast cancer again. Now that doesn't mean you can't have breast cancer, even if you've had your breast removed. It can happen, but it's way less likely. So that's the end of the first day. And you'll want to do the practice problems, any practice exercises that are on the Evolve site. And there will be maybe a practice quiz. And then we'll continue on with some more of this information on the second day of this week's work. See ya.